Okay, Ananya has joined. Good evening, Ananya. Good evening, Ananya. Who else has joined? Come on, fast. Say good evening so I know who else has joined, who everyone has joined. Okay, good evening. Good evening, Ananya is there. If you can hear me, you also tell me that you can hear me properly. Today we are going to start with the new chapter today, which is photosynthesis in higher plants. Okay, so we are going to start with that. Okay, Rahmat has joined. Rahmat, very good evening. Okay, so I am audible, I guess. I'm just waiting for everyone to join so that everyone can join, and I will start with the new chapter today, which is photosynthesis in higher plants. So we have completed transport in plants, and we are doing this plant physiology right now. So in this series, we are starting with the photosynthesis in higher plants today, and in this we will learn about some of the very important and basic and logical concepts in photosynthesis, how the photosynthesis occurs, what are the different phases, what are the different structures which are responsible for the photosynthesis, everything, each and everything. Wahid is also joined. Wahid, good evening. So yeah, good evening. Good evening, Bhuvanashri. So everyone just join fast, join very fast so that we can start off with today's session. And in this we will see how the plants, how this process is taking place in plants. So how in plants this process is responsible for the production of food, and how it enables the plant to produce their own food, that is glucose. Okay, and how the plant does that, what are the different phases and everything. Yeah, good evening Rohit, good evening. Okay, so I guess everyone has joined or everyone is joining fast. So we can start off. It is that all animals, including human beings, they depend. We all are dependency. The producers. When we talk about producers, these are plants. These are plants. Why they are plants? Because they are autotroph. Autotrophic. They are autotrophic. Autotrophic means they can prepare their own food. In very simple language, that is, they can prepare their own food. And any organism which is not autotrophic cannot prepare their own food. It is not autotrophic, it cannot prepare their own food. So what does that mean? That all other organisms which are not autotrophic, they depend upon these producers, these plants, for their food. They depend upon these plants for their food needs. Okay, Jennifer has also joined. Yeah, good evening, Jennifer. So they depend upon plants for their need. Okay, for their food needs. Okay, so the thing is, whether it is any plant which is eating any animal. There are some animals which directly eat these plants. Okay, so these animals, they are termed as herbivores. They are termed as herbivores. Okay, now there are some animals which are eating these animals, which are ultimately eating plants. Okay, so their ultimate source of energy plant is again plant, because they are eating an animal which ultimately eat plants. Okay, now if this animal is dead, Sushma has joined. Yeah, Sushma, good evening. So if this animal is dead, the bacteria and fungi will feed on it. Okay, bacteria and fungi will feed on it. So these are decomposers. So the decomposers, they are also feeding on animals, which is the ultimate source of energy of which this animal was also plant. Okay, so what I'm trying to say is that it doesn't matter, which level of trophic level you are, on which level of the trophic level you are, it doesn't matter. The only thing is which matter here, which is mattering here is that plants, they are the ultimate source of what? Food. They are the ultimate source of food. And what is the ultimate source of energy? It is sun. It is sun. The ultimate source of energy is sun, because sun is providing the sunlight, and the sunlight is used by these plants to prepare this food. So the ultimate source of energy is sun, and the ultimate source of energy, food, is plants. Okay, so now we will study how these plants, they are able to capture this sunlight. They are able to capture this sunlight from the from the sun, and they convert this light energy into food, into chemical. This energy is stored in the form of chemical bonds. This basically energy is stored in the form of chemical bonds. So in this chapter we will understand that. Okay, so see, all all animals including human beings depend upon plants for their food. Have you ever wondered where the plants get their food? Green plants in fact have to make, or rather synthesize, the food they need, and all other organisms depend upon their, depend on them for their needs. So that is what I was telling you, that all other organisms, they depend upon them for their needs, for their food needs. And green plants carry out photosynthesis. Photo means light. See, photo means light, and synthesis is to make. Synthesis means to make. So when plants make their food in the presence of light, or by utilizing the light, it is called as photosynthesis. It's a physico-chemical process by which they use light energy to drive the synthesis of organic compounds. And what is the organic compound? It is a glucose. It is glucose, C6H12O6. This is the main organic compound which is produced during photosynthesis. Okay, so to produce the organic compounds. Ultimately, all living forms on earth depend on sunlight for energy. As I told you, that ultimately the ultimate source of energy is sun. Okay, and the use of energy from sunlight by plants during photosynthesis is the basis of life on earth. Yes, this is the basis of life on earth, because life on earth can survive, can be there, can thrive only when there is energy. Only when there is energy, and the ultimate source of energy is sun, which in turn is providing this energy to the plants, and the plants, they are using this energy to convert it into food, and this food is provided to all other organisms which are present on the earth. Okay, so photosynthesis is important due to two reasons. First, it is a primary source of food of food on earth, and it is responsible for the release of oxygen. Yes, oxygen, which otherwise is called the byproduct of photosynthesis. Would you believe that oxygen, which otherwise is called the byproduct of photosynthesis, is a life-giving gas, oxygen. Okay, you know how much oxygen is necessary for us, for our survival. Okay, so the thing is, plant is utilizing the carbon dioxide and water. Plant is taking carbon dioxide from the atmosphere and water from the soil, and they are utilizing these inorganic substances to ultimately convert them into glucose, C6H12O6, glucose, and oxygen, and this oxygen is ultimately released into the atmosphere, which is utilized by us, for the purpose of respiration, okay, into the atmosphere by green plants. Okay, now this chapter focuses on the structure. This chapter, one second, just give me a second. So now coming, what do we know? what do we know? Now, let us try to find out what we already know about photosynthesis. Some simple experiments you may have done in the earlier classes have shown that chlorophyll, that is a green pigment of the leaf, light and carbon dioxide, they are required for the photosynthesis to occur. They are required for the photosynthesis to occur. You may have carried out the experiment to look for the starch formation into, in leaves. Okay, so you may have carried out the experiment to look for the starch formation in two leaves, a variegated leaf or a leaf that was partially covered with the black paper and one that was exposed to light. On testing these leaves for starch, it was clear that the photosynthesis occurred only in the green parts of the leaves in the presence of light. Yes, we have done all these experiments in our earlier classes, in our previous classes, in which we saw that the green parts of the plant, which is the leaf of course, it contains a photosynthetic pigment called chlorophyll, and in this chapter we are going to learn more in detail about chlorophyll. Okay, so we are going to do that. We will learn about it in more detail. Okay, so the thing is, we have done these experiments in our previous classes and we know the basics of photosynthesis. It is not something very new for us. We have done that. We are doing this from class 6th. Okay, so we have already learned a lot and we already know the basics of photosynthesis. In this chapter we are going to go into the depth of it, how this process is taking place. Okay, so we have done these experiments and everything. There is no problem in that. Another experiment you may have carried out is a half leaf experiment, where a part of a leaf is enclosed in a test tube. Okay, you remember these experiments, yes or no? First of all, tell me, do you remember these experiments or do I need to tell you again? I think you must be remembering all these experiments. These are very basic experiments that you have done in your previous classes, I think, and all of you must remember these classes, these experiments. Okay, so if you have any problem, you can tell me. I will again explain those experiments. There is no problem in that. Okay, I can also explain it, and if you know it, if you are like comfortable with it, then we can move further. So another experiment was there, in which it was called half leaf experiment, where a part of leaf is enclosed in a test tube. A part of leaf is enclosed in a test tube containing some potassium hydroxide soaked cotton. And what does potassium hydroxide do? It soaks or it absorbs carbon dioxide, while the other half is exposed to air. Okay, so what we are doing is, we are doing half leaf experiment. It looks something like this. Let me show you. This is a leaf. Okay, and half of the leaf is inside the test tube. It is inside the test tube, and this test tube has cork in it like this, and the leaf is pushed into this, inside this cork, and it has KOH. So half of the leaf is outside the test tube, which is exposed to the atmospheric carbon dioxide and everything, and half of the leaf is inside the test tube, in which the potassium hydroxide is put, and this potassium hydroxide will absorb all the carbon dioxide which is present inside the test tube. And now this leaf is exposed to sunlight. Okay, this leaf is exposed to sunlight. So when you do the starch experiment, after some time you will observe, yeah, you will observe that the part of the leaf which was inside the test tube, does not show you the starch formation, while the part of the leaf which was outside this test tube will show you the starch formation. Which means what? That carbon dioxide is necessary for the process of photosynthesis. Without carbon dioxide, photosynthesis cannot take place. So these were the experiments, basic experiments, which proved that light is necessary, chlorophyll is necessary, everything. So we have done these experiments in our previous classes. The setup is then placed in the light for some time on testing for the starch. The later in the two half of the leaves, you must have found that the exposed part of the leaf tested positive for starch, while the portion that was in the tube tested negative. This showed that carbon dioxide was required for the photosynthesis. Now we are going to do some more experiments. Some more experiments are given here in your in your syllabus. There are these experiments, so we are going to do these experiments, read about these experiments. They are very important, so pay very close attention. Okay, now see, there are some experiments that we are going to do. See, it is interesting to learn about those simple experiments that led to the gradual development in our understanding of photosynthesis. The first is Joseph Priestley, 1733-1804. In 1770, he performed a series of experiments that revealed the essential role of air in the growth of green plants. Pay very close attention to each and every word here, okay? So Joseph Priestley was there in 770. 1770. What he did was he performed a series of experiments that revealed the essential role of air in the growth of green plants. And Priestley, you may recall, discovered oxygen. He discovered oxygen in 1774. So Priestley observed that a candle burning in a closed space, a bell jar, soon gets extinguished. So what he observed was that a candle, when it is kept in a closed bell jar, it gets extinguished after some time. It soon gets extinguished. And similarly, a mouse would soon suffocate in a closed space. Yes, if you place a mouse in a closed space, it will also soon suffocate. So he concluded that burning a candle or an animal that breathed the air both somehow damage the air. Damage the air means what? That they are using some of the portion of the air, or they are using some part of the air which is essential for their life. Otherwise they wouldn't get dead inside this closed space, or the candle would not get extinguished. So but when he placed a mint plant in the same bell jar, he found that the mouse stayed alive. So now when the same experiment was being done with the keeping the mint plant inside this bell jar, now the candle kept on burning and the mouse remained alive. To his surprise, the mouse remained stayed alive and the candle kept burning. So what did he concluded from this? Though he hypothesized, Priestley hypothesized as follows, that plants restore the air whatever breathing animals and burning candles remove. So the thing is, he did not know at that time that what is this gas or what is the particular part of air that this mouse or candle they are using. He didn't know that. But yeah, one conclusion he or to he reached to one conclusion, or he hypothesized it as that plants are restoring that part of the air whatever the breathing animals and candles they are removing. Okay, so because he discovered oxygen in 1774, and these experiments were done in 1770, which means what? That oxygen was not discovered at that time, so he didn't know about oxygen at that time. Can you imagine how Priestley would have conducted the experiment using a candle and a plant? One second. Remember, he would need to rekindle the candle to test whether it burns after a few days. How many different ways like you think of to light the candle without disturbing the setup? Now, this is a question which is open to all of you. You can answer this question, like how it is possible to rekindle the candle without disturbing the setup. You have to rekindle the candle which is inside the bell jar, and how would you do that? So it's an open question to all of you. Let me see who can answer that. It's an open question. You can answer afterwards also. There is no problem. Okay, right now we will continue with the other portion. Using a similar setup as the one used Priestley, but by placing it once in the dark and once in the sunlight. Now there is another scientist. So you have to remember the names of all these scientists and what they did. It's a very important part of the chapter. A lot of questions come from this part as well. Okay, so pay very close attention. Using similar setup was used by a different scientist. Okay, and but what he did was, once he placed this same setup in the dark and once in the last light, and the name of the scientist was Jan Ingenhousz. Jan Ingenhousz showed that sunlight is essential to plant process that somehow purifies the air fouled by burning candles or breathing animals. So what conclusion did he reach? That the candles and the mouse, whatever damage they are doing to the air, that can only be repaired when there is sunlight. So sunlight is essential. Okay, sunlight is essential to the plant process, that whatever plant is doing, we don't know about that. He didn't know about that at that time. So he just concluded that whatever the plant process is doing, for that process to occur, sunlight is essential. Okay, So, somehow purifies the air, followed by the animals. Engine house in an elegant experiment with an aquatic plant showed that in bright sunlight, small bubbles were formed around the green parts of the green parts around the green parts, while in the dark they did not. So, what he did was he also took aquatic plant. He also took aquatic plant, and this aquatic plant placed in inside the water, inside the water. So, when this plant was placed in sunlight, there were bubbles seen in the green parts of the plant. There were bubbles seen near the green parts of the plant, so which means that some of the gas was being produced by the plant. But when the same setup was kept in dark, that is when there was no sun, okay, these bubbles were not forming. So he also, this also kind of gave him a confirmation that this process is occurring only in the presence of sunlight. Okay, so later he identified these bubbles to be of oxygen. Okay, and hence he showed that it is only the green part of the plants that could release oxygen. So you have to remember what what these scientists were doing at that time. You have to remember these points, that Jan Ingenhouse, what did he do? That he concluded that that the green part of the plants could release oxygen, and that is only in the presence of sunlight. Jan Ingenhouse. Okay, now moving on, it was not until 1854. Now another scientist has come here. Okay, it was not until about 1854 that Julius von Sachs, another scientist, Julius von Sachs, provided evidence for the production of glucose when plants grow. So, it was Julius von Sachs. He also provided evidence. That the plants, they are producing glucose when they are growing, and glucose is usually stored as a starch. His later studies showed that the green substance in the plant is chlorophyll, as we know it now, is located in the special bodies later called chloroplast. So, Julius von Sachs, he did a lot of work, and a lot of things were proved by him. A lot of things were explained by him. Whatever, what were the works that he did? That first of all, he provided evidence for the production of glucose. Okay, and his later studies showed that the green substance in the plant is chlorophyll. There is a green substance in the plant which is now known as chlorophyll, is located in the special bodies called chloroplast. Now it is called as chloroplast within the plant cell. So he kind of discovered this chlorophyll and chloroplast. So at that time, this name was not given to it, like chlorophyll and chloroplast. This is something we know it now, but at that time he was the one who first identified it, at least. Okay, yeah, there is green substance, or there is special bodies in which this green substance is present. Okay, and he found that the green parts in the plant is where glucose is made. So he also found that this glucose is made in the green parts of the plant. So this is also something which was done by Julius von Sachs. Okay, so now see, and the glucose is usually stored as a starch. Now we consider an interesting experiment. An interesting experiment was done by T.W. Engelmann. Okay, and using a prism. Okay, he did use this prism. He split the light into several spectral components and illuminated a green alga, a cladophora. So what he did was, he kind of separated the different spectrums of the light using the prism. Okay, and he illuminated with the green alga with it, and green alga taken by him was cladophora. Remember the name of the scientist, T.W. Engelmann. He placed in a suspension of aerobic bacteria. Yes, so what did he do? He took green alga, cladophora, placed it in the suspension of aerobic bacteria. Okay, now the bacteria were used to detect the sites of oxygen evolution. Yes, why? Because they were aerobic bacteria, so they will get accumulated at a place where there is oxygen, because they are aerobic bacteria. So they will get accumulated at those places which are oxygen efficient, which have oxygen. Okay, so these bacteria, they were used to detect the sites of oxygen evolution. He observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. Yes. So when he observed that where is this bacteria mainly getting accumulated, he saw that only in the blue or and red regions, the blue and the red regions of the cladophora which were eliminated, only at that places the bacteria accumulated. You know, mostly the bacteria accumulated in that region of the cladophora which were eliminated with the blue and red light of the split spectrum. So at a first action spectrum of the photosynthesis was thus described. So this is a kind of a first action spectrum, and don't worry about the action and this absorption spectrum. I will explain you this absorption and action spectrum in very detail, just so you know right now that first action spectrum, first kind of action spectrum was given by him, because he split, first of all he split the lights, he split the light into spectral components, so that all the spectral components of the visible light region were seen, and all the different regions were eliminated with the different split region of the spectrum. And he did observe that that it is the blue and the red light region of the spectrum in which the aerobic bacteria is getting accumulated the most, which means that which means what? Now see what it resembles roughly the absorption spectrum of the chlorophyll A B discussed in the section 13.4. So we will discuss it in the section 13.4. Don't worry. So by the middle of the 19th century, the key feature of the plant photosynthesis were known. So by the end of the 19th century, okay, the key features of the photosynthesis were known, namely that the plant could use light energy to make the carbohydrates, that is glucose, from carbon dioxide and water. The empirical equation representing the total process of photosynthesis for oxygen evolving organisms was then understood as. So at that time, the process of photosynthesis in the form of a chemical equation was understood as this. This was the empirical reaction. This was the empirical reaction, that is CO2 plus H2O in the presence of light giving CH2O, which is a carbohydrate, general empirical formula of a carbohydrate plus oxygen, where CH2 is represented a carbohydrate, example glucose, a 6 carbon sugar. Okay, so this is representing a glucose molecule. Okay. Okay, Sushma is asking and Shazia is asking, illuminated meaning, sir? Illuminated to supply or brighten with light. Okay, Shazia has explained the question of Sushma. So the question came from a student and the answer also came from the student. Very good. So a milestone contribution to the to the understanding of photosynthesis was that made by a microbiologist, yeah, Cornelius von Neil, yeah, a very important, a key scientist here, that Cornelius von Neil, who based on his studies of purple and green bacteria demonstrated that photosynthesis is essentially a light dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrates. Okay, so this Cornelius von Neil, he was a very eminent scientist. Okay, and he gave a milestone contribution to the understanding of photosynthesis. What he did was, based on his studies of purple and green bacteria, he demonstrated the photosynthesis is essentially a light dependent reaction in which hydrogen from a suitable oxidizable compound. So right now it was not clear at that time, from this line it is not clear from where the hydrogen is coming, so a suitable oxidizable compound, just so just at that conclusion, a suitable oxidizable compound, it is reducing the carbon dioxide to carbohydrates, that was sure. Now in green plants, hydrogen, H2O is a hydrogen donor. So now it came to know that in green plants it is water, which is donating the hydrogen, so it is a hydrogen donor. And one second. Okay, so in green plants, water is a hydrogen donor and is oxidized to oxygen. Okay, it is itself oxidized to oxygen and it is giving up hydrogen. to reduce the carbon dioxide. Some organisms do not release oxygen during photosynthesis. When H2S, that is hydrogen disulfide, okay, is used, it is used when H2S instead of a hydrogen donor for the purple and green sulfur bacteria, the oxidation product is sulfur or sulfate, depending upon the organism and not oxygen. Of course, when water was used, oxygen was coming, but when H2S is being used, it will give sulfate, sulfur or sulfate, depending upon the organism. Okay, hence he inferred that the oxygen evolved by the green plant comes from water, yes, and not from carbon dioxide, yes. This was a very big confusion at that time, that the carbon dioxide plus water both have oxygen, both have oxygen, and this was not clear, but that the first of all we are getting carbohydrate C6H12O6 plus we are getting oxygen. So we were not clear, where is this oxygen coming from? Is it coming from water or is it coming from carbon dioxide? But from his studies, it was clear that the oxygen during photosynthesis, it was coming from water. It was coming from water, not from carbon dioxide. You also remember this. This was later proved. It was also later proved by the radioisotopic techniques. The correct equation that would represent the overall process of photosynthesis is therefore that this is the equation. This is the balanced chemical equation: 6CO2 plus 12H2O in the presence of light gives glucose C6H12O6 plus 6 molecules of water and 6 molecules of oxygen. Okay, where C6H12O6 represents glucose, oxygen released in the from water. This was proved using radioisotope techniques. Okay, and now moving on, one thing is, note that this is not a single reaction. Yes, that this is not a single reaction. The reaction which you are which you are able to see, CO2 plus H2O giving C6H12O6 plus oxygen. This is not a balanced equation, but this is the overall equation. Okay, so this overall equation of the process does not tell you all the steps how this carbon dioxide is ultimately converted into glucose, ultimately reduced into glucose. It's a stepwise process. It is a multi-step process. It's not a single step process. In this equation, you see it in a single step, the carbon dioxide getting converted into glucose, but this is not a single step. It's a multi-step process which we will learn. So it's an overall equation. It's an overall equation, not the whole process. Okay, so see, but description of a multi-step process called photosynthesis. Okay, so it's just a description of a multi-step process. Now, where does this photosynthesis take place? You know the answer. It is chloroplast. Where is this photosynthesis taking place? It is taking place in the chloroplast. No. So now, starting with this, the structure of chloroplast, which is very necessary for all of us to know. Okay, so first of all, we will understand the structure of chloroplast. So see, you would of course answer that in the green leaf, or you may add that in the chloroplast, based on what you earlier read in chapter 8. You are definitely right. Photosynthesis does take in the green parts of the leaf, but it does also, it does so also in the green parts of the plant, in other green parts of the plants. So you would recollect from the previous unit that the mesophyll cells in the leaves have a larger number of chloroplast. Yes, if you recall the structure of leaf, if you have any idea of the structure of leaf, so see, this is the upper surface of the leaf. If we look at the structure of the leaf, this is the upper surface of the leaf. It is called upper epidermis. Then you have like these long and tall slender cells, okay, like this. These are called as mesophyll cells, and this is palisade mesophyll. This is palisade mesophyll. Palisade mesophyll. Okay, after palisade mesophyll, you have somewhat rounded and spongy cells with intercellular spaces like this. Okay, and these rounded spongy cells with intercellular spaces like this, it is called spongy mesophyll. It is called as spongy mesophyll cells. Okay, and there is the lower layer of the epidermis, which is called as the lower epidermis, and it has opening as well. This opening in the form of these bean shaped cells are called guard cells, and this opening pore is called stomata. It is called as stomata, through which the exchange of gases takes place. And this is stomatal aperture. This is stomatal aperture, and this is substomal space. Stomatal aperture and substomal space. So this is the structure of a dicotyledonous leaf. To be specific, it is a structure of a dicotyledonous leaf. Okay, in which the lower surface has a lot of stomata as compared to the upper surface. Upper surface has no stomata or very few stomata. Okay, so in this, the mesophyll cells, they have these spherical, these like oval, not spherical, like oval bodies called chloroplast. They have these oval bodies called chloroplast. And inside the chloroplast, we will see the different structures which are responsible for performing the different reactions of the photosynthesis. So we will see that. We are now going to see that structure only. So you have studied the structure of chloroplast in chapter 8. Within the chloroplast, there is a membrane system containing the grana. Now see the structure of chloroplast. See, this is the structure of chloroplast. You see this this is structure of chloroplast. Within the chloroplast, there is a membranous system consisting grana, the stroma lamellae, and the fluid stroma. So there is this stroma lamellae, the fluid jelly-like portion on which is acting as a ground substance, of all of this. This is known as this is known as grana. This is this is known as stroma. This is known as stroma, the ground portion. Okay, and these is small, these disc like structures, you can see that these disc like structures, like the coin like structure. Okay, these coin like structure, they are known as thylakoids. And the pile of thylakoids, this this pile of thylakoids, or it looks like there is a the pile of coins. It looks like the pile of coins. Okay, so the pile of coins, this is known as grana. So this is a structure of chloroplast, in which there is a it has a double membrane bound structure. The outer membrane is there, inner membrane is there. Connecting to grana, there is a stroma lamellae, called as a stroma lamella, or this ground substance is called stroma, and there are ribosomes as well. There is circular DNA as well. If you have studied the structure of chloroplast in chapter 8, you know that it contains a DNA, circular DNA as well. So the stroma lamellae and the fluid stroma is there, and there is a clear division of labor within the chloroplast. What does it mean that there is a clear division of labor? That the specific structures are there for the specific functions. They are performing their own specific functions. No one is interrupting into another functions. So the membrane system is responsible for trapping the light energy, and also for the synthesis of ATP and NADPH. Remember this. These are the products of light reaction. ATP and NADPH, these are the products of light reaction. And where is this occurring? The membranous part. What is a membranous part? Thylakoid. Because only thylakoid has the chlorophyll. The thylakoid has chlorophyll, and chlorophyll has the power of trapping the light energy. Okay, so this thylakoid, which has the power of trapping the light energy, it is a membranous portion of the chloroplast, and it is responsible for the light reaction. We will study the light and the dark reactions in detail. Okay, so this is responsible for the light reaction, the thylakoids. So this is occurring in the thylakoids. What are the products of light reaction? ATP and NADPH. Okay, now in stroma, the enzymatic reactions incorporate carbon dioxide into the plants, leading to the synthesis of sugar. So see, the different parts performing different functions. The thylakoid is performing the function of light reaction, in which the major products are ATP and NADPH, while in the stroma portion, that is the jelly portion, in which there is no chlorophyll. Yeah, do remember this. There is no chlorophyll in this ground substance, a jelly portion. It has enzymes which are responsible for incorporating the carbon dioxide into the plant and leading to the synthesis of sugar, ultimately producing what? Glucose, which in turn forms starch. The former set of reactions is directly light driven. Yes, since The former set of reactions, you'll see the set of reactions in detail, don't worry, and they are called as light reaction. They are called as light reactions because they are directly driven by light. They are directly taking place with the help of light, and therefore they are called as light reactions. The latter are not directly driven by light, but they are dependent on the products of light reaction. What are the products of light reaction? Again, remember, ATP, NADPH. These are the products of light reactions, and they are used in dark reaction. They are used in dark reaction. Since they are used in dark reaction, therefore, dark reaction is dependent on the products of light reaction. So it is not directly driven by light, but it depends upon the products of light reaction. Okay. So which are ATP and NADPH? Hence, to distinguish the latter, they are called as by convention the dark reactions. And by dark reactions, please do not get confused. It does not mean that it is taking place in the dark. No, it is not taking place in the dark. It is also taking place in the light. That is when there is sunlight. These reactions, these dark reactions, they are also occurring when the light reaction occurs and the products of light reactions are produced. So this will occur only when there is light. So dark reactions will also initiate when there is light. But why they are called as dark reactions? Because they are not directly driven by light. Okay, so that is one thing that you need to keep in your mind, that they are not directly driven by light. However, this should not be constructed to mean that they occur in darkness. What I was trying to tell you, the next line is that only, that it does not mean that they are occurring in dark, okay, or they are not light dependent. Yes, okay, so you should not get confused to understand that, since they are called dark reactions, they are taking place in night, at a night, or in the dark, and they are not dependent upon light. No, they are dependent upon light, okay, but not directly. Everyone got this point? Everyone understood it? Come on, first of all, tell me, till this point of the chapter, if there is any doubt, you can ask me. I am open for the doubt portion now. If you have any doubt, you can ask me. Come on. Okay, actually I was trying for trying for so long. The class was scheduled to take place from 8 p.m, but there was some technical error and I could not start the class on time. I tried to I tried for so long, but I could not do that. Okay, so but at last I had to take the class on YouTube. So but now I think the technical error has been resolved, and tomorrow I will take class on the app only. Okay, on the app only. Okay, but today it's late now. Okay, and I will not start a new heading here because it will take time to complete, so I will not start it. It does it does not help if you start something and do not complete it. So I will not start it. I will start only tomorrow now. Okay, for today I think it's enough. And if you have any doubt, if you have any problem, you can ask me anytime. Okay, so now everyone take care. Good night. We will now meet tomorrow and start with the new topic, which is how many pigments are involved in the photosynthesis, and we will learn about the some of the pigments which are involved in the photosynthesis. Is that clear to everyone? Yeah, okay. You're welcome, everyone. You're welcome. See you tomorrow. Okay, bye bye.